This session is chaired by Dr. Alexander Tropsha. I'm thrilled to introduce him today. He is the K.H. Lee Distinguished Professor at the UNC Eshelman School of Pharmacy, UNC Chapel Hill. He has authored or co-authored over 300 peer-reviewed research papers, reviews, and book chapters. He's an elected fellow of the American Institute of Medical and Biological Engineering, also a co-founder of Predictive LLC, a UNC startup specializing in building models and tools for chemical toxicity prediction. He is a co-organizer of this workshop and helped us put this workshop together. Thank you so much, Alex. The floor is yours.
Thank you, Serena. I'll be very, very brief. I want to start by thanking all speakers for describing and highlighting the importance of data and databases that can be used for CAD applications. In this session, we will hear from 4 leading experts in chemical data analytics using both available and synthetically feasible molecular libraries and methods of computational chemistry, machine learning, AI, and physical modeling. And the first speaker of the session is Alexandre Varnek, who is a full professor of theoretical chemistry, the head of the Laboratory for Chemoinformatics, and the director of the Masters of Science program in Chemoinformatics, one of very few in the world, at the University of Strasbourg, France. He's also a PI of the Chemoinformatics groups at ICReDD at Hokkaido University in Japan, published 3 books and over 300 research articles and book chapters. He's an editor-in-chief of Molecular Informatics journal. He has been organizing highly popular workshops in chemoinformatics in the last 15 years in Strasbourg. And he is the recipient, congratulations, Sasha, again, of the 2024 Skolnick Award from the American Chemical Society. So, Sasha, you have the floor.
Okay. Thank you very much, Alex, for this kind introduction. It's a great pleasure and great honor to give a talk on this prestigious meeting. So I'll speak about specific methods of exploration of large chemical spaces. So nowadays chemistry generates a lot of data, and some 10 to 9 molecules are currently recorded in public databases, and more than 10 to 26, up to 10 to 26 are stored in the proprietary databases of large pharma companies. The motivation to create such large chemical libraries is based on the assumption that the library size correlates with the number of hits in visual screening. This, however, is not obvious because both library size and chemical diversity must be considered. The question arises how to find the library that is the most suitable for a particular molecular design campaign. In this talk, I would like to present you molecular cartography or chemography approach, which is promising solution to the preparation of made-on-demand libraries, and also in combination with modern AI techniques, it may help to design novel molecular entities. Chemography considers data distributions on 2-dimensional maps characterizing both structures and properties of molecules. How such maps would be obtained? In fact, a molecule can be represented as an object in n-dimensional chemical space. In order to obtain the map, we need to reduce this dimensionality to 2 dimensions using specific dedicated methods. There exist many various methods of dimensionality reductions, and certainly the data distribution vary as a function of the method applied. By some reasons, we selected a method which is called Generative Topographic Mapping, or GTM. This slide briefly resumes the GTM algorithm. So first, from large chemical library, we select a rather small representative data set for which we generate descriptors, and then the algorithm inserts the flexible sheet called the manifold This manifold flexible sheets is inserted in the data cloud, and its position approaches the high density areas. The manifold unbending results in two-dimensional map on which all compounds are projected. Each projection is fuzzy. It means that each projection is described by the probability of the molecule location in different nodes of a rectangular grid superposed with the map. Such projection looks like a spot of ink smeared across a sheet of paper. Accumulation of such fuzzy projections results in data density distribution, as you may see in the right upper corner of this slide. And in turn, this density distribution can be encoded by the vector of probabilities calculated for the nodes of the grid. The vector length is equal to the number of nodes, which is user-defined parameters. Thus, instead of consideration of individual objects, GTM can consider their probability distribution. The probabilistic nature of GTM opens an opportunity to use this approach not only for data visualization, but also structure property modeling tasks. With the help of color code, generative topographic maps can accommodate some information about various molecular properties using such called GTM landscapes. One can consider three basic types of landscapes: density landscape, characterizing the data density; a class landscape, characterizing population of activity classes, active, inactive, in different areas of the map; and activity landscape, which reports an average activity value in each node of the map. Each of these maps can be encoded by a vector characterizing distribution of chemotypes and activities in the chemical space. Due to the fact that GTM landscapes characterize the probability of data and activity distributions, they can be used for activity predictions. Thus, for any new molecule projected on the map, a class landscape can be used to predict a class of the compound. It means that it works like classification model. The activity landscape works like a regressor or regression model to predict the activity value. And if the compound is projected in a low density or empty area, it is considered to be out of the model applicability domain. GTM can efficiently be used to analyze chemical space of individual libraries. As an example, here we consider the Campbell database containing some 2 million diverse molecules annotated with various properties and biological activities. On the density landscape, one can easily identify high density zones which can be associated with some chemotypes. Biological activities can be mapped on GTM forming activity landscapes, some of which are shown on the slide. Each landscape accommodates the molecules studied against particular biological targets. We built more than 700 activity landscapes for chem data, and for any new compound projected on the map, one can identify the most similar compounds in Campbell and predict its pharmacological profile for more than 700 biological activities using these activity landscapes. The next part of my talk concerns pairwise library comparison, in which one can identify structural motifs unique for a given library. I will show you a few examples, and the first example concerns pairwise comparison zinc database containing commercially available compounds with Campbell collecting biologically relevant compounds. Altogether, more than 1 billion structures were considered. So our goal was to identify non-overlapping part of zinc containing chemotypes never been biologically tested and which could potentially enhance screening libraries. On the other hand, non-overlapping part of Campbell containing chemotypes missing in the commercial space would be recommended for the enhancement of biologically biased commercial libraries. Starting this project, we have discovered a typical problem of cartography of big data. A map resolution may not be sufficient to separate different compounds classes. Thus, a map shown on the slide accommodates a subset of more than 3.5 fragment-like compounds from zinc and Campbell. In order to retrieve the chemotypes, we needed to identify the zone populated exclusively by compounds only from one database. These are red zones for zinc and black zones for Campbell. But we observed on this map mostly some green zone populated by both classes, which means that GTM manifold is not able to identify the existing library-specific zones. In order to identify pure zones, a technique of hierarchical maps has been applied. For each selected mixed zone, we built a new child map in which the two classes, two databases are better separated. If this separation is not sufficient, we continue with new child maps. And in this example, we see on the level 2 of zooming, this child map accommodates only 400, a bit more compounds, already has zones which are populated by only by zinc or Campbell compounds, from which we can extract particular chemotypes, maximum common substructures. So, and in such a way, we built more than 40,000 hierarchically related maps, which allowed us to extract more than 100,000 chemotypes never been biologically tested. These chemotypes from zinc, and more than 20,000 chemotypes missing in the commercial chemical space. And we developed a tool allowing user to navigate in this chemical space atlas containing so many individual maps. So another example concerns the reshaping of proprietary collection of Boehringer Ingelheim company. And this collection contained some 2.2 million compounds. One day, the company intended to diversify its library by purchasing compounds from Aldrich Sigma Market Select database, containing more than 8 million of compounds, and the budget, certainly, as usual, was limited. So we have superposed Sigma and Boehringer compounds on a GTM, from which we selected unique maximum common substructures from Sigma, which corresponds to more than 400,000 molecules, which then were filtered using Lipinski paints. So we also used GTM activity profiling as a filter. And in such a way, we selected some 1,200 compounds which were recommended for the enrichment of the proprietary collection. And another example concerned recently delivered Freedom Space. This is a combinatorial library of 5 billion drug-like molecules. And in this study, we demonstrated that Freedom Space is not redundant, but rather complementary to well-known real space containing 40 billion structures. Indeed, GTM built on a relatively small representative subset of 1 million each, shows very little overlap between freedom and real spaces. So you can see a clear frontier between black and red zones. And then when we look at the landscapes, so we can state that the freedom database is enriched by more drug-like QED index is in average high for freedom compounds compared to real space compounds. Okay, let's now consider multiple libraries analysis. Here we compare one reference library with a series of, let's say, external libraries, And the goal is to select the library the most similar to the reference one according to similarity score. In this study, we considered DNA-encoded library as external and Campbell as a reference database. So just to remind you that DEL represents a combinatorial collection of small molecules covalently attached to short DNA tag. A DEL results from 2 or 3 steps chemical reaction between selected building blocks. Using collection of commercially available building blocks and selected reaction rules, one can generate thousands of DELs containing billions of molecules. And it should be noted that the synthesis and screening of DEL is pretty expensive. Therefore, selection of the optimal DEL for particular drug discovery campaign is an important chemoinformatics task. So, using the fact that each library can be represented by a vector, we can easily suggest a metric, for example, Tanimoto coefficient, which can assess the overlap between these 2 databases, CHEM and one of DELs. And then we performed a computational experiment using a collection of 79,000 of building blocks from a molecule database with the help of eDesigner tool prepared by WILEY company. We designed 2,500 DELs having size from 1 million to 1 billion. From each of these DELs, we selected a representative data set of 1 million compound. So altogether, we considered 2.5 billion compounds. So then we intersect each DEL with Campbell, and then we selected the highly scored DELs. So the best DEL, so we discovered that the best DEL selected in such operation already cover more than 50% of chemotypes of Campbell. And then we were looking for the DELs which they cover the best this gray area which corresponds to Campbell, and red area corresponds to zones populated by Campbell. So and in such a way, we discovered that already 3 DELs covers more than 80% of Campbell chemical space. But then when we projected all DELs on this map, we discovered a gray area, the area populated of NP compounds which are not covered by the DEL chemistry. When we consider ensemble of the chemical libraries, we can benefit of the fact that each library could be represented by the vector. So we can consider the space of chemical libraries in which each data point is an individual library. And for this ensemble, we can build another GTM, which we call meta GTM, which this GTM characterizes relationships between libraries in this space. This is an example, so in which we plotted the 2,500 DELs with respect to Campbell. The Campbell is positioned just in this on the bottom left corner, and red star is characterizes the most closest DEL in chemical library space, and the gray star is a remote database. As you look at if you look at the density landscape on the right, you can see that the landscape of the closest DEL is the most similar to density landscape of Campbell than density landscape of the remote DEL. And then we can certainly use color code, for example, to analyze reactions which were used to build these DELs, and we can conclude that most of DEL space is covered by coupling-based libraries, and very few DELs were created with heterocyclization reactions. Okay, next topic which I would like to discuss concerns application of chemography in combination with AI tools to generate novel molecular structures and chemical reactions. So the idea is very simple. So let's consider the tool like autoencoder, and we can use the vectors of the latent vectors to build GTM. Then we can build a activity landscape from which we can automatically or manually select an area populated by useful structures, for example, active compounds from which new structure could be sampled. So we applied this approach several years ago, and we discovered new entities enriched with the novel structures enriched with new scaffolds. Then we applied similar approach to generate new unseen chemical transformations, new unseen variants of Suzuki coupling reactions. So for this purpose, we use chemical reactions stored in USPTO database, the reactions extracted from the patents. So as a first step, we transformed all chemical reactions in a condensed graph of reactions with some sort of pseudomolecule, this technology which is developed in my lab. So then we built a GTM. This map has been colored on the latent variables, and we selected some zones populated exclusively by Suzuki coupling reactions from which new transformations were sampled. So, and we generated in such a way 13 new, with respect to the training data, reactions. And then, so we did perform we did validate this discovery by ourselves, but we found then 5 of these new reactions in SciFinder, which is much larger than USPTO. And then, so we extended this approach on the chemical space built on any variables, not necessarily on latent variables of autoencoder. So for the training, we used both SMILES and molecular descriptors, any descriptors. And then as soon as the model is trained, we just use as input molecular descriptors, and the neural network outputted as the SMILES chemical structure. So this technology can be efficiently used for inverse QSAR tasks. So now I would like to speak about cartography of really ultra large, billions molecules. The problem with conventional pipeline, which includes enumeration, standardization, descriptor calculation, and projection, is that it is pretty time consuming. So certainly the time varies as a function of the molecular descriptor used, but for, let's say, Morgan fingerprints, we had, let's say, for 1 million compounds for projection, we spent for one processor 12 hours, which is pretty long time. And therefore, we decided to build a neural network which skips enumeration step and significantly 10 to 3, 10 to 4 times accelerates the preparation of the map. So I would like to give you details of this neural network architectures. It is given in this reference. We already this paper in ChemArchive. But this is just a illustration how it does work. So this is the model can be built on a relatively small set. Already 25 compounds is sufficient to train a model predicting GTM for 90 million DEL. And you may see the maps built with a neural network and map which is practically identical to the map built for enumerated chemical structures. So finally, why do we prefer GTM compared to popular TSNE or UMAP techniques? So recently we performed a benchmark related to neighborhood preservation. That means we compared the neighbors in the initial space and in the latent space of the map. So we used for this purpose more than 100 sets extracted from Campbell's, 3 different descriptor sets. And so results are also in ComArchive. And this slide actually resumes this extended benchmark study. So instead of, in sense of neighborhood preservation for a given data set, so all 3 nonlinear techniques, TSNE, UMAP, and GTM, they are practically at the same level. So certainly they never have 100% neighborhood preservation. The best, it depends on the fingerprint, is 50 or 70%. But GTM clearly outperforms other methods when we project on the map new compounds. So that's why our choice of GTM is well funded. So very last slide. So I would like just to summarize all advantages of GTM. It considers data probability distribution, which makes it suitable for big data analysis. When we have a lot of data points, we need to go from individual projections to data distribution. First, so the GTM model can be built on relatively small representative subset, and then it can accommodate new data. The landscapes can be used for regression or classification tasks. What is extremely important, the data collection can be encoded by a vector which enables fast comparison of libraries. And finally, coupling of GTM with the AI technologies facilitates de novo design. So I would like to thank my collaborators who participated in this work and our industrial and academic partners. And I would like to thank you for your kind attention.
Dr. Varnek, thank you very much for excellent representation and analysis of GTM and what it can do with chemical libraries. There have been a few questions on the sidebar, which I'm going to read some to you. Some you've actually answered as you progressed. So they have been addressed. So one of the questions that I'd like to select is that you've showed that Zinc database showed better SAR. Why did you use Campbell as a reference library for DEL? If you could comment on that.
Yes, you know, so we, in fact, we consider, let's say, we consider a task of selection of DEL, the best DEL for primary screening. And for primary screening, when the target is not known, so we need to have, as a reference, the most diverse data collection. That's why, simply. So we consider Campbell as a diverse data collection.
Thank you. Any comment on whether and how you could use GTM to address the questions about activity cliffs and chirality cliffs?
I would say that GTM is not. We try to we have a work with Jürgen Bayrat on this topic, but GTM is not the best tool to identify activity cliffs for sure, because GTM works with the, let's say, the average data distribution, and it captures mostly this continuous landscape.
Thank you. And the last question, given the time constraints, when you compare GTM with others, the descriptor set or representation that you use is 2D, but bioactive compounds are 3D. Can you comment on 2D versus 3D original representation in the context of activity?
We published several papers on protein conformations, on chemical reactions, network, etc. when we use specific 3D descriptors, and GTM can give very interesting, let's say, cumulative view on large MD trajectory of proteins or a series of related chemical reactions. So it can be efficiently used for visualization and analysis of complex 3D structures using specific descriptors.
Okay, thank you very much. If you have time, could you please answer some of the questions and the Q&A online? If you kindly could do that, Sasha, and thank you very much for an excellent talk.